Good evening, everyone. Students, am I audible?
Yes, ma'am.
Okay. Is the screen visible clearly? Is the screen visible?
Yes, ma'am.
Okay. So today we'll be discussing about population genetics, and later on we'll be discussing about the gene concept. That is small topic. So I kept it in last. Okay. So let's start with the population genetics. So as the topic tells you, like what it is a study of population. Okay, till now what we have started is like we are taking some plant, we are doing something, we are studying about the individuals. Okay, like one particular individual, how the trait will be inherited and how the progeny will look like. If the individual is made with another particular individual, then what is the progeny constitutions will be, what is the gametic constitution will be, we are studying about that. But you see, when we are going for the inheritance pattern study, we are not only a particular individual, we are interlinked. In a society we are interlinked, right? Like our parents, they belong to two different families, their parent, they belong to two different family, their parent, they belong to two different family, like that with because of all the interlinkings and
Bang
marriages.
Zigolo, sabon.
Hello.
Hah? Patunyim Zigolo. Okay.
Okay,
Upon the
so
Klingon.
what we are doing here is,
Okay,
by the time
Zigo.
generation progresses, am I audible clearly, students? Hello.
Some background voice is audible, ma'am, along with yours.
Yeah, I can also hear that, but I'm not. Wait.
Otherwise, mute all participants, ma'am.
Okay. So, with the help of all the marriages and matings, the population gradually goes. Okay. And what is a population basically is, it is a group of interbreeding individuals. Okay. They can, which can freely intermate between them, means which has the ability to freely intermate between them. Okay. What does that mean is, they belong to mostly because here the reproductive parameter is set between one population and another population. It is like in one particular population, they used to belong to a same species first of all, because within species only intermating is allowed invariably. Means if it is a monkey, then it can mate with a monkey only. If it is a plant, means if it is a particular plant, let's say rice, it can freely intermate with another plant, another rice plant only. Okay, between species intermating is somewhat successful, but not always. And especially if you talk about the population genetics, in case of populations, in case of animals and plants, they can mate randomly also, but that does not exist in case of human population. So we have a very kind of structured population. We humans used to select the progenies, and how human is to select how the their parentage and with which family they are going to interlink, and how the family lineage will go on. Okay, so that's all about the population we are going to study. And why the study of population genetics is important, it is like, if your grandparents or their grandparents are in having some diseases, there is a possibility that through your one of the parents or both of the parents, that particular gene will be inherited to you, and that will express in some day or in some form. Okay, So because and other aspects are also important. I just gave one important aspects of population genetics over here, why we need to study. Apart from that, whether the particular individual is going to be a good one, or heterozygosity will be high or low, inbreeding depression will be there or not, all these things are related to, means how the progeny will be, how the progeny performance is going to be. All those things are linked to your study of the population genetics only. So let's begin with it. So starting with few terms, like gene pool. This is a common term, and what it tells you is it tells you about the common all the sets of sum of the all total of genes and all the alleles available in a population. Okay, I told you in a population you make it very clear that what is a population. Population is a group of individuals that coexist within a certain space at a time and capable of interbreeding with one another. This particular phrase is very important, capable of interbreeding with one another. So like I told you about the population, so gene pool, when we are talking about a gene pool, we are talking with respect to a particular species. Okay, and in that context we will be somewhere in the future lectures we will be dealing with the gene pool concept in detail. But for now we are just talking about the population concept. So population concept is nothing but the group of individuals that coexist within a certain space. Okay, so all the species belonging to same species coming under primary gene pool. All the species belonging to same genera, they will be constituting the secondary gene pool. Here in the primary gene pool, there will be 100% fertility if you cross two individuals within a particular species. But if you'll cross the individuals between two species, that the fertility level will be somewhat fluctuating, and we can encounter some of the less fertile or sterile kind of progeny. Okay, tertiary level is when you are going out of the genera, that is intergeneric or outside the particular and very distant relatives. That particular case is tertiary gene pool. And the fourth is quaternary gene pool, that is a new gene pool introduced. That one is sometimes called as ocean group also. And that's all about the your gene pool, and we'll be studying somewhere when we'll start dealing with the plant breeding section. Okay, so that was what gene pool. The next term is gene pool is constituted of various genetic information, like all the alleles will be present in alleles are the alternative forms of gene, as I already told you in previous classes. So let's say take a for a particular gene, that is let's say this is the gene, that is a gene is responsible for your body color, let's say. Okay, so it is governed by two alleles, capital A and small a. So here this is the particular gene is A, means the gene is your color gene, skin color, and skin color is governed by either capital A is going for one allele, that is capital A, that is for fair color. In normally in color of skin, you see dark color means black skin is dominant over white skin, so we'll be going with that notation only. Capital A is that is black skin, and your small a is fair skin. Okay, okay. So what they're telling is genotype frequency. What is the genotype frequency is in a particular population, how the population will be constituted of some individuals, isn't it? In a population, some individuals will be there XYZ something something. Let me show you a picture. Let's see here you see there are several frogs. Okay, if you take the skin color, then let's say three frogs are there. If you take the skin color, they constitute one population, isn't it? So this frog will be having a particular genotype, this will have another genotype, this will have another genotype. Let's say this one is having capital A, capital A. Since this is darker in color, this is capital A, capital A. This is small a, small a. And this is capital A, small a. Whenever we are talking about genotype, means I am talking about the diploid condition. Okay, diploid means an organism's somatic cell condition I'm talking about, whether it is diploid or polyploid. So for simplicity I'm taking a diploid case. So in case of diploid, you can find any somatic cell, two alleles are there. So that two allele can be anything, can be capital A, can be small a, or can be capital A for a particular gene of skin color, isn't it? So in the population you will find here, for simplicity there are three frogs, but let's say in this particular case, there will be some number, let's say 1, 2, 3, three number of this capital A. Green, this green one, that is small a is 4 in number, and intermediate one is 2 or 3 in number. Okay, so they constitute the number of times the particular allele, the frequency. Okay, so proportion I showed, like for total 12 individuals, total 10 individuals were there. Out of them, 4 individuals were having smallest capital A genotype. Okay, then 3 were having small a, and rest 3 were having capital A small a. So if somebody is asking you like genotype frequency, so it is like out of 10, 4 individuals are this particular genotype. Out of 10, 3 individuals are this type. Out of 10, 3 individuals are like this type. So you can say it as 0.4, 0.3, 0.3. So genotype will be 4 by 10, 3 by 10, and 3 by 10. That is genotypic frequency. If the next term tells you that like allele frequency, here we are talking about the proportion of a specific allele. Allele means here we are referring to either capital A or small a. So proportion of any specific allele in a population. So how many times the capital A allele is there in the population and small a allele is there in the population. So for any particular case where two alleles are there for a particular gene, there can be three types of situation, right? So capital A allele will be there in two number of copies in case of your genotype capital A small a, because it is capital A. Okay, because here both the alleles are capital A type, and it will be in case of your capital A small a, it is present in one copy, isn't it? Capital A small a. So whenever we are going for counting the frequency of capital A, we have to consider capital A along with capital A small a. So if the total number of capital A number of individual is 4 in number, and they have two alleles each, so the frequency of this will be 4 into 2, that is 8, and this particular allele will be 1 into 3, that is 3. Okay, so here it is 8 plus 3, that is 11. So total 2 alleles are there and 10 individuals are there. So out of 20, so they will have a total 20 alleles. Out of 20 alleles, 11 alleles are there which is having what? 11 alleles are there which is your white in color. Okay, means which is representing the capital A. Likewise, you can calculate for small a also. Okay, so whenever we are counting the total number of individuals over here, we will be calculating it like total number of dominant, means when we are calculating for dominant allele, total number of individuals of dominant allele into 2 plus half of heterozygous, that is capital A small a, that is your allele frequency. Okay, forget about all this thing, you just remember the definition from where you might be asked any question. Okay, okay, then speciation, another term is speciation. Speciation is the process of formation of new species. That is the process of your courses like crop breeding and all, you would of Oryza, like we have 20 species of Oryza glaberrima, Oryza this, that means their species, how the species is arising actually. So from one parental species, there will be some sort of mutation or some sort of variation will be created, and because of their geographical partitioning or something will occur, so that one the parental species will diverge into two different species. They have something different with them, means they have some characteristic, either the leaf color will be different, either the seed shape will be different, or something, some characters are different, so that they occupy a particular niche, they particular a particular habitat, and their ecology becomes somewhat different, so a new species is created. So this term speciation tells you about formation of new species. Then these two terms, that is genetic drift and gene flow, will be discussing in detail. For your definition point of view, I just put it here. That is genetic drift is random change in allele frequency over time. So this is a random phenomena. Like you can compare this with your a random phenomena, other random phenomena like mutation. So where randomly means your any change in your nucleotide sequences occur. Okay, so what will be occurring here in the genetic drift is, here the allelic frequency was in this particular population. Here allelic frequency was something like out of 20 alleles, out of 20 alleles, only 11 alleles were capital A type. Sorry, 11 were capital A type and 9 were small a type. Okay, so here what about the other thing is, let's say in this particular generation some sort of selection and something occurred, or some individual died. Okay, after that what will happen is there will be less number of this small a's. I think out of this particular thing, let's say four of the individuals died with small a genotype. So what will happen to this particular population? Now that four of the individual died means eight alleles of the small a are lost. So only one allele is remaining for this particular thing, that is small a means only one allele representing this particular small a allele will be remaining in the population. That might be in the form of capital A small a or something like that. This was just for your example sake, means how the genetic drift is occurring. So initially the population was having some other kind of composition, but because of something, some random change, some sudden phenomena, your allelic frequency will change. That's what the genetic drift tells you. Then gene flow is means what is gene flow is, there are two particular different populations. This is one population, this is another population. If some alleles are getting, means if some individuals are passing here to here to here, means they are moving from here to here. So, or if particular individual of this particular population is getting is mated with this particular one individual of this particular population, so the genes present over in this particular population will be increase in possible alleles between the population, because whatever
Your
is
voice
present
is
over
getting
here,
cut,
there is
ma'am.
a chance that they might be present over here. So because of intermediating between them, because of different regions, there is a quick case that there might be a okay. So what I am talking over here is, here the gene flow will be there occurring between the two population. Okay, so that will alter the frequency of the alleles in both the population. Okay. Then next is inbreeding. Inbreeding means the mating between the relatives or mating between the individuals which have the common ancestry, means which are related genetically, means intermating between the population. individuals, they are belonging to same family. In layman language, if I'll tell you, mating between the individuals who have the common parents in some generation. Okay, so inbreeding, that is what called inbreeding, and inbreeding causes increase in the homozygosity. This thing you need to remember, like inbreeding, what it will cause, because they have the similar ancestry, so some of the genes they will be common and they will be sharing some of the genes. So because of inbreeding, because of the mating of the related individuals, homozygosity will be increasing. Homozygosity means either the constitution of capital A or small a, that is the condition, right? If from both the parent capital A alleles will be coming, means if in the case that both the parents will be sharing the same alleles, so there is a chance that we will be getting higher amount of homozygotes, that is capital A or small a. Okay, then gene pool I already told you. Next term is genetic equilibrium. Genetic equilibrium means change, no further change in the allele frequency. This does not, if in the terms of population genetics, I'll tell you. In the next slide, we are going to deal with the law. There, I'll tell you in detail about the genetic equilibrium. And here, what they're telling is gene pool in which which is not changing the allelic frequency. Means whatever allelic frequency, let's say 0.6 or zero point 0.4, capital A out of one means out of total. Your out of total allele, let's say it is one, means frequency is taken as one. So out of that, a zero point 0.4 is the frequency of your recessive allele, that is small a. Zero point 0.6 is the frequency of your dominant allele, that is capital A. Okay, so in this particular generation it is like this, in the next generation also it will be like this, and the next generation also it will be like this only. If that condition is maintained, that is called as genetic equilibrium. Okay, and if the condition is there that this genetic equilibrium is established, there is nothing, no, none of the genotype is altering, and your allele frequency is constant, that means that it is not evolving. How this statement is correct, I'm going to tell you. Okay, so here another term is the random mating. So what is random mating? Random mating is a condition where there are in the all these individuals, let's say here 10 or 15 individuals are present. Okay, this one, anybody. So random mating condition is that any individual in a population has equal opportunity to mate with another individual. So there is no specific that these two individual will mate, these two will mate, or these two will mate, these two will mate, or if the 10 individuals are there, two will form a pair and they will mate. Nothing like that. Any individual can mate with any individual. This is possible in animals and plants. This is seen, but not in the 100%, because natural selection is operative somewhat on all the population. Okay, this is the ideal condition, but still I'll be telling you the concept what it is. So this, if you take this particular individual, this can, the chances that it will mate with this particular individual is same as that of this one, this one, this one, this one. It can mate with anybody. It can also mate with anybody. That means all the population, all the individuals in a population can mate with anybody at random. Okay, there is no specific like okay, this one is green and this one is green. These two will mate. Nothing like that. Okay, so that one is random mating. So because the mating is random, we can expect that whatever the alleles they are carrying, they will be, means all the genotypes will be produced. It is, if we just specifically choose something, like you have some rice plants, you will just select, you will just choose the tall plants and you will mate among them. Can you expect in the progeny that dwarf plants will arise? No, you cannot expect that. So here in the random mating, since all the individuals are freely mated, means they are allowed to mate with each other freely, so you can expect that here when the mating is random, so they're telling that they have taken two types of genotype, that is paternal gamete and maternal gamete. Okay, so here they have taken that to the example, like heterozygous individual capital A and small a, and paternal gamete here, and here the case is like capital A here the means the population, you see what is the composition of the population. This population is having three genotypes: capital A, small a, okay, then small a. These three types of genotypes are present over here. And these three, this one, any of the individual can mate with each other. This one can mate with this one can mate with this, any of the individual can mate with each other. So in any of the cases here, out of this population, if they made in random, this is not, I'm not talking about a particular mate, so I'm just talking about mating. What is the possibility, right? Here how they are calculating the frequencies of allelic frequency is that your maternal gamete, that is, let's say capital A frequency is P and small a frequency is Q, means Pa plus Q. Here I have taken the total frequency, like P plus Q equal to 1, means total frequency will sum to 1, or you can tell that it is in the frequency terms we are telling it as 1. Of you, if you tell in the percentage basis, out of 100%, P and Q in the percentage of your particular allele, means let's say it is 0.6 or zero point 0.4, okay, 0.6 and zero point 0.4 means it is 60% and this particular thing is 40%, so they are totally constituting 100%, okay. So this frequency of P and Q, P is the frequency of capital A and Q is the frequency of small a. And again the paternal gametes will be capital A and small a type in this particular population, isn't it? So the P, if it is mating with this capital A, means capital A is mating with capital A only, so the frequency will be P into P, because P is the frequency of capital A, and it is mating with the individual, that is paternal gamete is capital A, so it will be P into P, that is P square, isn't it? Then if the capital A is mating with small a, it is like P is the frequency of capital A and it is mating with the paternal gamete, that is small a, which is having frequency small q. So P into Q, that is PQ. Likewise, if the small a is mating with capital A, it will have Q into P, that is PQ, and Q into Q. If it is mating with small a, means Q into Q, that is Q square. So this is the frequency of the alleles that is present in particular population, okay. So here the population will be having the frequency of this will have different values in different diallelic which is a basic law in your population. So what it is like, in an open random mating population, you just say words. In an open random mating population, under the circumstances of no selection, mutation, migration, or random drift, the generation, the frequency of the alleles or the genotypic frequency will remain constant from one generation to the next generation. It was proposed by Hardy and Winberg separately. Okay, so we'll be discussing this principle, what it is. Okay, so let's begin with it. Here it is that the population showed you, that is P square, 2PQ and Q square. This is the composition of the particular, this is the frequency of your alleles, that is P and Q, and P square, 2PQ and Q square, this is the frequency of the particular genotype, isn't it?
Ma'am,
That is the case. So Hello.
Unable to see your screen, ma'am.
Okay, just a second.
Lot of problem with the signal, ma'am. Poorly. Exit అయిపోయిందా?
Is the slide visible now? Is the slide visible now, students?
It's loading, ma'am.
Hello.
Ma'am, it's still
Okay,
loading.
it will come. Not coming.
No, ma'am. Yes, ma'am, it came.
It came.
Yes, ma'am.
Okay. So the frequency that is p square, 2pq and q square, that is the genotypic frequency. So this represent capital A, this represent capital A, small a, and this represents the small a, small a. Okay. So the Hardy-Weinberg equilibrium tells that this frequency that is p and q, this will remain constant if all the conditions are met, and this frequency that is p square, 2pq and q square, this will also remain constant. So that is the condition. So if that particular conditions are met, then in that particular case, p square plus 2pq plus q square, this will be 1, equivalent to 1. Okay. And along with that, p plus q, that is your both the allelic frequency, they will also sum up to 1. If the Hardy-Weinberg equilibrium assumptions are made, all the conditions are made, like there is nothing, no selection, nothing is there made, in that condition, you can tell that these two cases will be there. Okay. So let's see what are the assumptions of Hardy-Weinberg equilibrium. I know this topic might be a little bit boring, so I have put some pictures for means so that you can understand it better. So here, let's say, start with the first principle. That is, there is no selection. There should not be any selection. Means all the individuals should have equal amount of fitness to stay in the population. Okay, no individual is selected open, and there should not be any sort of mutation. Mutation should be absent. Mutation is any alteration, any sudden change in your any sudden heritable change that is occurring at your DNA, which is causing some sort of variation. There is no sort of mutation. Mutation should not be present. So this is having two eyes, so it should have two eyes. No mutation is allowed. No migration means if they are residing in this area, they should be residing in this area only. They should not pass on to any other area. So that you can see here there is some variation between the population present over here and here. There should not be any migration. They should not move from here to here or here to here. Okay, that the population should be sufficiently large. Okay, so there should be, if the population is sufficiently large, means there is less chance of inbreeding between them, and that is another condition. And there is condition that there is random mating, full random mating should be there. Okay. uh, and random mating, I already told you, all the individuals have equal opportunity to mate with all the individuals present in the particular population. And this large population plays a big role. I'll be telling you in the next section how it plays. So this is the 5 rules you need to remember. So that is the description. And one thing is that this Hardy-Weinberg equilibrium, I told you, no, Heisenberg equilibrium. Heisenberg principle tells that this is the equilibrium condition. That is, this p square 2pq q square p plus q is equal to 1. And this is, both the conditions are 1, and this particular p and q does not change over the population, does not change over generations. This is the condition called where whatever allelic frequency I told you, like zero point 0.6 or zero point 0.4, if it is like this, it will remain like that only over generation. It will not change. So that is called as Hardy-Weinberg equilibrium. Equilibrium means it has allelic frequency p and q is equal to 1. So this is the condition. Never confuse that equilibrium might be like 0.5, allelic frequency p is equal to 0.5 or q equal to 0.5. That is not the equilibrium, means that is not the condition of Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium tells that in frequency can be anything, but this has to remain constant over generation. That's what the principle tells. But the 0.5 is the condition that is called the equilibrium frequency. But that is not the frequency dealt directly. That is the not only frequency dealt with the Hardy-Weinberg principle. Hardy-Weinberg principle is applicable at any combination of the P and Q. Anything can be there. But that is the only condition has to meet is it has to remain like that over generation. That's what the principle tells. So never confuse with that. Okay. Okay. So, then what are the phenomena that will cause the allelic frequency change? Here are the, I have given the definitions. I already told you, so I'm just skipping it. Then starting with the mutation, what will be the mutation? Like here you see, this is the normal DNA. This is the one basis. From here, suddenly something happened, and here there is one deletion. Like something has got out of this particular thing. Okay, so in that particular space something else is inserted which is not previously present. So this will cause some sort of changes. So this is also a type of mutation. Here something was there and in the next generation this was lost. So this is also a type of mutation. Here it is insertion, here it is deletion. Here first deletion has occurred, then insertion has occurred. Here totally deletion has occurred. So because of this mutation, it means after the initially it may be condition like it is small a allele and there was another allele like capital A. After this particular deletion, there might be a case that this particular condition may give rise to another form of allele which is a mutant allele, which may be anything else like A dash. Means since it is derived from A, most of the conditions are same like A only, but it will be given as something else. Okay, let's say three alleles like P, Q, R, or something like that. So no, we should not take P, Q, R here. You will confuse with the allelic frequency. Let's say A1, A2, A3. Okay, so let take a gene that is A. It has three allele. It has two allele A1 and A2. A1, A1. This one previously another one will be there. That is A2. Okay, this is A2. But because of the mutation in both A1 and A2, somehow two new alleles are generated. That is, this might be A3 and this might be giving rise to A4. So in that particular population, initially total one, the total frequency one was divided in only two things. But here, because two new things have arisen and they will be occupying some of the portions. So now the frequency out of this total frequency, they will be occupying some frequency. So this condition is arising. It will means originally if say let's say this condition, if the mutation is arising, it will means originally if say let's say out of 10 individuals 6 were like capital A and 6 4 were like 6 were like A1 and 4 were like A2. So we used to call it as 0 0.6 and 0 0.4. But now that one of the A1 has converted to A3, one of the A2 has converted to A4. So now the number of A1 will be 5 in number, it will be 3 in number. So the initial frequency 0.6 and 0.4 has changed to 0.5, zero point 0.3, zero point 1, zero point 1. So the frequency of A1 and A2 has now changed. So we cannot call this as Hardy-Weinberg equilibrium because the equilibrium frequency, because the frequency is not constant over here. it has changed in the next generation, isn't it? So that in this manner, it is causing the alteration in Heidelberg equilibrium. Okay. The next thing is migration. Migration is movement of individuals from one particular location to other particular other locations. So you see here in this particular location, only this blue birds were there. You see the big. This is blue. These blue birds are there. Here in this population, there is no blue bird. All the blue birds were capital small in that genotype, and all the pink birds present over this population are capital A in genotype. Suppose because of some reason the blue bird just flew over to this new location, and it has intermated with one of this individual. So here first thing is this new allele is introduced, this small allele is introduced, and here only the homozygous small was there. So if it will intermate with any of this individual, new combination will arise, that is capital A small a, which was not previously present. So it has disturbed the equilibrium frequencies over, means Hardy-Weinberg equilibrium over here also and here also. So this is the case of migration. So migration can be E migration or I migration, means the movement of either some individual may be coming into population or they may go out of the population. Okay. So this is the here only because of this migration the gene flow occurs between two populations. Like from here one individual went to this population and it will intermate, and after intermating there is some gene in this new individual is carrying some genes of this blue bird also, this green bird also, a pink bird also. So this here the gene flow has occurred. We'll call it like gene flow has occurred. Okay. So next thing is like natural selection. See here one bird is coming. This is a frog eating and fish eating bird. So if it will choose to, if it can choose anyone to eat, isn't it? But if it will choose only the black ones to eat, so gradually the black frog population will go on decreasing. So this will disturb, means initially two of the black and two of the green were there. If it will only eat the black one, after eating black one, only one black frog will be remaining. So out of three, it is one. Then again, another black will go, means there is no black frog left in this particular population. So initial frequency of 2:2 means two green and two black population has now shifted to only 2:0. So the frequency has changed, isn't it? So in this particular population, this will also, this selection, that how the nature is acting upon, this is a case like um means just for illustration I have given this bird and the frog. But chosen by the nature, and they can breed and maintain their popular progenies, the inferior individuals will eliminated from the population. Okay. So in that, if that occurs, means the recessive alleles will lost from your population. So recessive allele may be completely lost from your population, that might be also a case. So natural selection is not, if it is operating, means Hardy-Weinberg equilibrium is not possible. So for Hardy-Weinberg equilibrium, this should not be there. Okay. So here in natural selection, three terms you need to know. Don't go on the graphs, just you see this particular three boxes. Now, what will happen out of the stabilizing, directional or disruptive selection? Here in stabilizing selection, the individuals with trait value that is in moderate region, they are favored. In the moderate region, individuals are favored. Average expression trait of individual is favored. So that is when this particular individuals are favored, that is called stabilizing selection. When we favor the trait of a particular extreme, that is called directional selection. Let's say in the population of selection for tallness, we will only select the tall individuals, or we will only select the dwarf individual. So in that particular population, we are favoring only one extreme. So that is called directional selection. And in case of disruptive selection, we are selecting the both the extremes. Like if for tallness selection, we will be selecting both tall plants and dwarf plants. Extremely tall and extremely dwarf plants are selected. Okay. So that is what here it favors the both the extremes. So these three terms, they might be asking you. Just keep this in mind, these particular terms and what they will give rise to. That's it. And here another illustration of your natural selection. Here the bird is choosing only to eat the green color ones, not the brown ones. So gradually the brown one will only remain and the green one will be lost from the population. So this will initially 1, 2, 3, 4, 5 brown and 2 greens were there. So if it will green or it will eat all the green bugs, then only the brown bugs will be there and no green one will be present. Okay. So nature acts, there should not be this like condition. Then only your equilibrium, how do you remember equilibrium is possible. But that is totally ideal case. That is not possible in anywhere in the nature. We can achieve somewhat nearer condition to that, but we cannot achieve the exact condition. Third thing is genetic drift. Genetic drift is like means suddenly if anything happens, sudden random phenomena or any catastrophic event is happening, because of that your gene frequency is drastically reducing. Okay. So that is also random change, means anything can happen to any of the particular population. So you see here many beetles are there, blue one and red one. We are just trying to kill some beetle with this particular beat, this particular bat, that is we used to kill the beetles. So if we just put it in random, it may kill anything, no, it may kill some red ones or blue ones, but here you see some blue ones are getting killed. So here it is not intentional that we will only kill the blue ones, but somehow with the stroke, with our stroke of the bat, only some of these were killed. So gradually what will occur, here they are 5 equal in frequency, but now you see, since these four of this blue were died, and now the population, there will be different, means the green one will be only, the blue one only, so they are double their number, and only one blue was remaining, so it has doubled to 2. So the frequency of that is 5:5 has changed to 10:2. So there is a drastic change in the gene frequency. Okay. In this one generation because of some random chance event. Then there are two important terms concerning this genetic drift. One is bottleneck effect and another one is founder effect. What is that? Let's see. First one is founder effect, means this was one population. Okay. This is some island. This is some island. They have been separated by some of the, this blue region indicates some water body. Let's say some sea. Okay. This is, let's say your Antarctica. This is your Australia. Okay. So they're far apart and they're separated by some virtue. Some of these individuals just, means some of this your butterflies, they just passed from this particular island to this particular island because of some reason. So here there was nothing, no individuals are there previously. Some of them just flew to here. So this has disturbed this particular population's Adirondack equilibrium, because some of the individuals have migrated to another place. So here the population frequency has been disturbed. And what will altered and disturbed, and what will happen to here in this particular case, here there is no previous individuals of this particular species. So whatever individuals went here, they are only few in number, and they are having random alleles. Nobody can tell that they will come, two of the capital A, two of the small a, two of the capital A small a will come. Nobody can tell like that. So randomly one heterozygous, two homozygous dominant have come here. So here there is no small a. You can see here. So because of this region, the allelic frequency, this particular allele is not at all present in this population, and the allelic frequency is very random and very narrowed down, isn't it? So this particular condition where few individual has to pass to new environment and they colonize in that particular area. So there you can find that very, from them only further breeding will occur and they will be giving rise to population. So because they are closely related, these two are totally same and this one is only a little bit different. So there will be high inbreeding between them because they are similar, and there will be less allelic frequency variation. So this condition is called founder's effect. So you can see here the pink and the green balls are there. Randomly they have selected, means here to another thing, this one is going, or randomly we have selected from other population, this one is going, or randomly we are going for this. So from here, um, this green one is higher in frequency, but after some generation there might be a condition like green ones are not favored. Here the pink ones are gradually increasing and green ones are lost. Gradually there might be a condition like this also. So anything can happen because this population is very small. No, there is very variety in this population. So next thing is bottle neck effect. Okay, next thing is bottleneck effect. Bottleneck effect is like um, whenever only small, because of any catastrophic event, let's say some calamity has occurred, like landstorm has occurred, or your landslides has occurred, or earthquake has occurred, so all the individuals in a area have died. Some of them somehow survived. So let's say whatever population survived from this particular thing, this particular event, so they'll be having only this particular thing and only one. So survival this one is bottlenecking event, that is your catastrophe. And this catastrophe is like let's say earthquake, and out of this total population, only this much population is surviving, and here only one of this green one is present and four of this purple one is present. So here the initial frequency was something different. After the catastrophic event, the frequency has changed. So if this condition is there, then also Hardy-Weinberg equilibrium is changed. The allelic frequency is changed, and hence the Hardy-Weinberg equilibrium is disturbed. Okay. So this is your bottleneck effect. Why it is called bottleneck effect is something only can survive, means bottleneck is narrow portion, right? So here all the things cannot pass freely. Something only at a stretch, only something can pass. All the things cannot pass. So that's why it is resembled like bottlenecking. That's why it's called bottleneck effect. For next thing is like non-random mating. Here what they're telling is don't go in detail for this. Just remember the terms like associative and disassociative mating. Associative means here they will be similar looking individuals or similar in genetic content. That is, if they're looking similar, then it is phenotypical associative. If they are genetically similar somewhat, that is genotypic associative mating. Disassociative means mating between individuals which are not similar. Okay. So if the similarities means if they are not similar in their genetic background, that is called genotypic disassociative mating, means they are completely different. If they are phenotypically, if they are mating between the phenotypically different organism, that is called phenotypic disassociative mating. So here it is non-random mating. Why? We are choosing something. We are mating. We are knowingly creating some mate pairs, and we are mating. We are allowing them to mate only, not the all individuals to mate freely. So this is a type of non-random mating. Okay. So that's all about your population genetics. And here in your exams they don't ask you about the all the um, problems, and also I'm just not discussing all those things. So these terms like gene pool, gene frequency, allelic frequency, population, genetic drift, random mating, gene flow, equilibrium, all those terms you need to remember in this. You forget about this slide for your understanding. I told you don't worry about this slide. Here in the Hadamard principle, the principle definition means the principle statement is important, and all the words I told you like large, open, random mating population, the allele and gene and genotypic frequency will remain constant. That definition is very much important. And they may ask you like what are the assumptions. So all these terms are important, like no selection, no mutation, no migration, random mating, all these things. So these are the important assumptions. So next, let's see. Here, what are the assumptions? So 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 here, what are the important, like stabilizing, directional, and disruptive. Keep this in mind. The next thing is your bottleneck effect and founders effect. The difference between these two is bottleneck effect will come when there is any catastrophic, the change in allelic frequency that or the genetic drift because of any catastrophic event. Then it is called bottleneck. But founders effect is some of the individuals, because of some reason they have migrated to some other place and they have created the new habitat over there, where the particular species is not present. There is no catastrophic event present in that particular phenomena. So that is the difference between these two, and these two phenomena you need to remember and understand. Okay. And next is you just need to go through these terms. That's it. Okay. That's all about your population genetics. And any doubts, students?
Ma'am, in gene pool, you told tertiary gene pool, quaternary gene pool, no, ma'am.
Yeah.
Other names.
What was your doubt in this?
After tertiary, you told quaternary, no, ma'am.
Yes, yes.
Other name for it, you told something, ma'am.
Super gene,
Super.
ocean gene, ocean group. I told ocean group is like it is called not super gene. Sorry, it is called ocean group. Ocean means sea. So sea means any sea. Ultimately, all the rivers are flowing into the ocean only. No, so there anything can be harbored. So that ocean group means tertiary gene pool. We used to write it like
Ocean group.
ocean group, and we used to mention it like 4 degree means quaternary. We used to mention it, don't denote it like this. And what
Okay,
is
ma'am.
comprises of in the tertiary gene pool we are talking about only with the particular individuals related genera. Up to that
Yes,
it is
ma'am.
limited, tertiary gene pool.
Yes,
But in
ma'am.
the ocean group or quaternary gene pool, sorry, it is not tertiary, it is quaternary. Fourth group is quaternary, which is ocean group. Tertiary is up to your intergeneric level. Okay.
Yes, ma'am.
In the quaternary, we are dealing with all the individuals, whether it may be a plant or animal. Basically, it deals with the transgenics, where we take the genes from any microbe and we are incorporating into any plant or animal for our benefit's sake, where we
Okay, ma'am.
modify any metabolic processes and all. So there is no restriction with the use of any particular parent with the other. If the mating
Okay.
is not successful also, we are doing it in vitro condition. We are doing it with the tissue culture. We are applying various biotechnological tool, genome editing principles, or various new developed tools to make that happen. Okay, so that particular thing is quaternary gene pool. Okay.
Okay, ma'am. Thank you.
Yeah. Regarding population genetics, there is not much for you for the study in the exam, but whatever definitions I told you, definitions are very important. Okay. If you find any doubt, then you can ask me anytime. Then moving to next concept, this is small topic which was related to your previous class actually, so I put it in last. So this is gene concept. Like from here they used to ask questions like this. This particular slide is very important. Like how the gene concept has evolved. Previously they used to think like in Mendel's time, they used to think like gene is nothing. Gene is a factor, something, or Mendel has thought that something is a factor present in the individuals which is governing your phenotype, the trait phenotype, and which is giving two extreme types. He didn't know whether the gene is present in the chromosomes or not, and nothing. He just told that there is a factor which is controlling a particular trait. That is Mendel's time. After that, Archibald Garrod has given the term. After that, I told you in chromosomal theory of inheritance that your Certain and Burberry has proved that chromosome means the genes are present over the chromosomes, right? So then they used to then they thought that okay, genes are present, and how the gene is like a beads on the string, means genes are arranged. Genes are some beads on the chromosome. Genes are some stretch up chromosome, and they're continuously arranged, and one gene cannot be subdivided further. So one gene has to do one certain kind of function. So what one particular gene can do, on that various scientists have given their theories. Okay. According to their experiments. So first one was Archibald Garrod. he has given in this one is important. He has given this particular concept in phenylketonuria, you remember, and he has given the concept of one gene, one metabolic block. What this means is one gene will give rise to one metabolic block. Here, the in phenylketonuria, one particular enzyme is non-functional. I forgot the name of the enzyme. It is useful in the phenyl. It will actually used in the utilization of your phenylalanine synthesis and its metabolism in the human beings. And that after this particular disease occurs in the individual, the ketone bodies actually gradually accumulate. Okay, so that particular gene is involved in the creation of that particular disease. That's what he thought, and it is actually involved, but it is involved in one of the pathways actually. But that time he thought that it is involved in the particular disease, so no need to bother about the disease. You just remember that the Garrod has given this concept, and he told like that gene is responsible for the total metabolic reaction, and because of that particular gene this disease is caused. So he told that it is one gene, one metabolic block theory. Okay, then Beadle datum in 1940, he told that one gene will give rise to not the whole metabolic block, but it will give rise to an enzyme. Means one gene is responsible for production of one particular enzyme, not a total pathway or any metabolic block. So somewhat refined theory this was. And again in 1957, Ingram has told that one gene is responsible for formation of a particular polypeptide, not even an enzyme. Enzyme is consist of several polypeptides, but one gene is not even responsible for all the polypeptides present in one enzyme. Okay, so one enzyme might be constituted by the gene product of several genes. Okay, not one gene. So that was a modified concept given by Ingram. Okay, and in recently Yanovsky has given the term that gene is the unit of recombination. I'll be telling you about gene is in one polypeptide they used to have frequently in the exam, and this one is very important. Okay, then as I told you, like classical concept of the gene, here they used to think that gene is the unit of function, and recombination, mutation, whatever will occur within two genes, all these thing can occur. Means two genes can recombine within each other. Two in one gene mutation can occur in the whole gene is responsible for mutation. Gene cannot be further divided. Gene is the smallest unit which we can call. Gene is the smallest unit which we can call, and we cannot subdivide it. Like you see in case of your physics, we used to call like first you used to study like a molecule. Molecule has several atoms. Atoms are previously they used to think that atoms cannot be subdivided. Again they come to the conclusion that okay, atoms can be subdivided into your electrons, protons and neutrons. They are the constituents part of the atom. Okay, so based on the particular time and their science level, extent of the science and study, they used to give the hypothesis. Okay, so here this was very classical in the early 1900. They used to think like that. After several experiments, and I told you, no, they used to think that the genes are arranged in a linear order like beads on a string. Like this is a string which is compared with the chromosome, and like this your genes are present. Okay, like this. And each gene occupies a specified position called locus. If the position of the gene changes, then character changes. Means everything, all the traits has to be related with your gene. And gene, the whole gene is responsible for something. Whatever function it is doing, the whole gene has to be involved in it. But later on the concept has changed. How the concept has changed means, you see, this is the particular this is not the correct illustration, let's say. Okay, in this particular gene, this particular stage of gene, then another scientist, that is Benger, he has done experiment in case of your R2 locus. Just for your knowledge sake, I'm just telling it one line. Okay, so in a virus he has done the experiment, and he came to know that he has seen the intragenic recombination. Intragenic recombination means previously we used to think that recombination is possible within two genes. But what he has observed is, he has observed the within one gene, let's say this is a particular gene, within here to here, recombination event has occurred. Okay, so he wondered like how this can be possible. So he did a very detailed experiment, and he came to know that within gene can be subdivided, and gene is, the whole gene is not the unit for anything, neither function nor mutation or non recombination. Okay, so gene can be further subdivided based on which particular action we are talking about. So based on that, they have given the terms like recon, muton, and cistron. This term, three terms are given by Banger itself. So what is the let's start with the cistron. Cistron is formation of the peptide, that is the protein. Part of the gene is going for the functioning, and the section of the gene, that can, the part of the gene which can independently go on its function. Let's say this is the stretch of gene. Okay, this is the total gene. If this particular gene is capable of independently going for its function and forming a peptide, this particular part can be called as a cistron. And one gene might be carrying several number of cistrons together. Okay, it is not that one cistron will be there in one gene. So part of a gene which is capable of independent function, that is a unit of function, that is called as the cistron. Or it is capable of synthesizing a polypeptide. Functioning means what is the function of the gene? It will form a peptide, it will form a RNA, and ultimately it will form a peptide, that is your protein. Then next thing is recombination. Part of, again, this is the whole gene, this is the whole gene length. The smallest part of the gene which can independently go on recombination with another part, that is called as the unit for recombination, or recon. And that is the concept here. Then third thing is muton. Muton is the smallest unit that can undergo mutation. If this is the whole gene, the smallest unit where we can induce a mutation, or which can independently go on go on and acquire a mutation, that is called as muton. Fourth, this is the new term actually. Initially three terms were given, these three terms, and complon was given later on. Complon is the unit of complementation. That is the smallest part of, that is a part of your gene which can go for complementation. No need to bother about complementation, that is not important for your examination point of view. So just remember the term, that is complon. Then, as I told you, Samuel Benzer has given these three. Then another thing you need to know, that is cistron will be somewhat bigger. It can have 100 nucleotide pairs, and even further also. Recon can be one to three nucleotide, like that. It may go for 1 to 2 pairs. Okay. Then muton is only smallest unit that you can introduce a mutation is one base pair. Means in any particular, in a ATGC thing, you can introduce mutation, that is the in any of the genes, right? So this can alter. So any particular base can change. So smallest unit of mutation can be one base, but smallest unit of recombination means it has to it has to change with somebody else. No. So the basic one is within two nucleotide, one nucleotide can go for recombination with the adjacent one or something like that. So for a minimum, it has to go for two nucleotides. And cistron is somewhat longer. Why? Because it has to carry some coding regions, which will be functionally coding for continuously some of the amino acid, which will be your protein sequence, and which will be a functional protein after all. That's why cistron is the biggest one. This is the biggest unit that can undergo mutation. This is the then next concept is split gene. This concept was given by Sharp and Roberts. The scientist two names is important, remember. And what the split gene concept tells you, that previously they used to think that all the genes are in a stretch and they're continuous. But this concept tells you that genes are not continuous. The genes there are two concepts like exons and introns. Two parts are there. So you can see the exon is the coding part, Exon is the coding part. Coding part means this will go on and form the functional protein. What this particular section is called intron. Intron is the non-coding part. They will not know form. They will not form any sort of protein. So protein coding part is exons, which is the functional part, and these are non-functional parts, non-coding genes. A non-coding section of a particular gene. So these exons are interrupted in between by introns. So gene is splitted. Gene is splitted into various codings and non-coding region. That is the concept of split gene. And when we'll be dealing with your transcription and translation, we will be dealing with these things in detail. Okay. So coming to parts of a gene, I told you, like gene will have a promoter sequence or poly site. All those things might be new to you. So we'll be dealing with them in detail. So for now, you just see for a particular gene, this promoter site helps you to for transcription and all those things to bind and for transcription and translation to occur. And these exons are coding sequences, introns are non-coding sequences, and these are the polyadenylation sites present over here, which are present in the mature mRNA and which are useful for its stability and all those things. So we'll be studying this section in detail in case of your transcription. And this is somewhat extended version of the previous table. Here it talks you like these two scientists are extra over here. They have given the molecular and evolutionary gene theory and nominal gene theory. But these three scientists are very, 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 very important. You need to remember anyhow. But you can go through these two scientists also, like Griffith and Newman. They have given molecular and evolutionary gene theory, and Griffith and Sloss, they have given the nominal gene theory. Sorry. So that's all about today's discussion. Okay. Any doubts, students?
Ma'am, split gene concept was given by
Sharp, SHARP.
and
Robert,
Robert, ma'am.
ROBERT.
Thank you,
Okay.
ma'am. Mm.
Okay. We'll be dealing, means all the exon intron, how they are affecting the thing. We'll be dealing with that in transcription and translation classes.
Ma'am,
Okay.
how many questions will we get from genetics, ma'am, in AFO?
Actually, I have gone through last 6 and 7 years questions. Okay. In your mains exam, all the agriculture sections are asked. And I found much of the questions from agronomy, soil science, and all those things. And from the genetic sections, I found 2 to 3 questions are there. Okay. Not much, 2 to 3 questions are there. So, like, which of the section I'm telling you like important, or sir is telling you like important, you need to focus upon them. And for your understanding, once when you are watching the video, means either in the live class or in the recording, so because in the letter means in the next time, next few months, you will not be getting that much time to see them. If
Ma'am, your voice is getting cut, ma'am.
hello.
Yes, ma'am.
Yeah. For the sections, means whichever I am highlighting, you need to go through them. And the rest of the portions you can skip before exam. No need to go through them. No problem.
No.
And that is for your understanding. Whenever you watch you found any doubt, you can go through your that video again and watch it, so that it will be clarifying your concept. Genetics is like such a thing, because if I'll just go on telling the facts, no, means you will not understand anything what is going on. And remembering the facts is not possible, because you have to go through several subjects, isn't it? That's why I have given some of the concepts. Hope you like the concepts, and it's okay for now. For the examination point of view, whatever terms and the scientists are somewhat important, and they will not ask from very depth, so I'm not going in depth also. For understanding little bit of your explanation is necessary. That's what we are giving. Okay. So any doubts, other doubts, students? I think no. And another thing, yesterday also, today also, and some in previous classes also, we have noticed that very few students are attending live classes. I know some of you are in fourth year, some of you are in third year or something like that. You might be busy with something, your lectures and all, but still, whenever you find time and you find time for scheduled classes, try to attend it. And if you are not able to attend it, sincerely follow, means whichever day the classes are finished, on that day itself or the next day you follow all the lectures, so that you will not they will not become piled up for to watch in the later times. Because if they get piled up, it will be very difficult for you to capture all the things at a time, isn't it? So do follow that thing, and discuss with us for any of the problems. Okay. So that's all about today's class. I'm ending the class now. Thank you all for your patient hearing.